Vahidam and welcome to my YouTube channel. Today we are going to discuss in detail about the nucleic acids, which is part of biomolecules and which is also part of CSS and PMS syllabus. So nucleic acids are not directly mentioned in the CSS PMS syllabus in the part of biomolecules, but they do are the part. So we need to discuss that. And basically we have studied about the nucleus. Nucleus contains nucleic acids, so it has an association with that. So we need to discuss about the nucleic acids as they are biomolecules or they are biomacromolecules. As far so far we have discussed about the carbohydrates, proteins. They are the biomacromolecules. So they have the monomeric unit. So the nucleic acids do have. What is the monomeric of nucleic acids? The monomeric unit of nucleic acids is basically nucleotide. But we are going to discuss about the both perspective. Nucleoside and nucleotide. Nucleoside basically is a nucleoside. A nucleoside consists of nitrogenous base and a sugar, while nucleotide consists of a nitrogenous base, sugar, and phosphate groups, one or more phosphate group. So we can say that there is only a difference of phosphate group between the nucleotide and nucleoside. So this is one of the basic difference between nucleoside and nucleotide. So we know that there are two types of basically nucleic acids. One is RNA and another is DNA. RNA is referred as ribonucleic acid, while DNA is referred as deoxyribonucleic acid. So what is the difference between both of them? Ribose nucleic acid and deoxyribose nucleic acid. From the name depicts that there is sugar in the RNA which is ribose in nature, and there is sugar in the DNA which is deoxyribose nature, that is without the oxygen at a certain position. We are going to discuss it in detail. So RNA and DNA have the basic unit as a nucleotide. So first we have to discuss about the structure of nucleotide. Okay, what are the components of nucleotide? There are three components of the nucleotide. Number one, that is nitrogenous base. Okay. Number two, a 5-carbon sugar. And number three, one or more phosphate group. So here I have drawn two types of nitrogenous base. One is pyrimidine and other is purine. So single ring containing nitrogen atoms are known as pyrimidine, and double ring nitrogen bases are known as purine. So I'm good. This is pyrimidine. This is single ring structure. This is purine, which has double ring structure. So we are going to discuss in detail that pyrimidine are of three types. One is uracil, other is cytosine, and this one is thymine. Uracil, this is cytosine, this is thymine. Okay, purine nitrogen bases are of two types. Number one is adenine. This one is adenine, and this one is guanine. So there are two types of purine bases. Purine is double ring structured, while pyrimidine is single ring structured. Purine are of two types, that is adenine and guanine, while pyrimidine are of three types: uracil, cytosine, and thymine. So here I have drawn the structure of both of the bases. Purine generalized structure, one type of purine adenine. So you can see that in purine bases there is difference of two groups between adenine and guanine. Rest of the structures matches the general structure of purine. And here is also the same concept in pyrimidine. There is a little bit difference between the bonding. So you can see the first difference in uracil, that is carboxylic acid group here. Here is amino group. Again, here is oxygen group. So there is a difference at first position. So there is nitrogen only. There is nitrogen, hydrogen, and there is nitrogen, hydrogen. So there are minute difference between all three bases. So pyrimidine have three types of bases, which is uracil, cytosine, and thymine. We need to know that these bases are complementary to each other while they are bonding. So we are going to discuss in detail about them. So these are the bases. Okay. This is I have drawn ribose sugar, which is the second component. First component nitrogenous base. Second component sugar. Sugar in DNA, in case of DNA is deoxyribose. In case of RNA is ribose. So this is ribose sugar. This is 5 carbon sugar. Carbon 1, carbon 2, carbon 3, carbon 4, carbon 5. So this is 5 carbon sugar, which is ribose in nature. What is the difference between ribose and deoxyribose sugar? The difference between ribose and deoxyribose sugar is that there is no oxygen atom at second carbon. Only hydrogen exists. So this has become deoxyribose sugar. So this is the only difference between RNA and DNA. So first component of the DNA RNA is nitrogenous base. Second component of the DNA RNA is sugar, which could be ribose in case of RNA and which could be deoxyribose in case of in DNA. So there is only difference of oxygen atom at position second. So the third component, okay. Now I am going to here stop you that nitrogenous base and sugar when attached together, that may be adenine. Adenine may attach to sugar. That may be guanine attached to the sugar. That may be uracil attaching to the sugar. That may be cytosine attaching to the sugar. That may be thymine attaching sugar. This basically form a nucleoside. So what I have defined, nucleoside consists of actually a nitrogenous base and 5 carbon sugar. Nitrogenous base and a 5 carbon sugar. So this is the carbon 5 carbon sugar. When it attaches to adenine, it forms one type of nucleoside. This is adenine nucleoside. So when this sugar attaches to the guanine, it forms guanine nucleoside. Okay, then there is a third additional component is that a phosphate group. When one or more phosphate group attaches to the nitrogenous base, ribose sugar and the phosphate, then it forms a nucleotide. These are the basic differences between nucleotide and nucleoside. So nucleotide is actually the basic unit or the monomeric unit of basically DNA or RNA. Okay, how does that attach? When keep it in mind when basically there is a glycosidic linkage between the ribose sugar and the nitrogenous base. So at first carbon there is attachment, so it forms N glycosidic linkage. When phosphate attaches, so there when nucleotide link together, the bond that exists between the nucleotide is phosphodiester bond. In basically nucleoside we have in an glycosidic linkage. In case of nucleotide we have phosphodiester bond. So this is how I have explained the differences between nucleotide and nucleoside. So further moving on to the structure of nucleotide, I am going to discuss here. So I am going to discuss here when nucleoside combines with phosphate group, which is P double bond O and others CH2 or negative. So when nucleoside attaches the phosphate group, it forms basically nucleotide. And up till now, 3 phosphobases can attach to the nucleoside, so it forms nucleotide. Suppose it's an adenine nucleoside. So what will it form? Adenine 3 phosphate group, adenine with the ribose sugar 3 phosphate group, it forms ATP, adenosine triphosphate. When there are 2 phosphate groups, it will form adenosine diphosphate. When there is a single phosphate group form. It formed adenine monophosphate. So these are the terms that I have to, when if suppose it's not adenine, it's guanine. So guanine will form GTP, guanine triphosphate, guanine diphosphate, guanine monophosphate. So GTP, GDP, AMP, like that. UTP, uracil triphosphate, UDP, uracil diphosphate, UMP, uracil monophosphate. When a person dies and his body attains the configuration of rigor mortis, that no organ is present. So there is actually the loss of ATP, adenosine triphosphate. As we know, adenosine triphosphate is also known as the energy currency molecule. So this is how the nucleotide and nucleoside different. Now moving on to the basic topic with nucleic acids, which are actually present in the nucleus of the cell. So we are going to discuss in detail. Number one, what is a nucleic acid DNA? The second one, RNA. Okay. DNA, deoxyribose sugar. It has ribose sugar. Okay. Okay. There is one other difference of nitrogenous bases. Thymine base is present in DNA. While uracil base is present in RNA. So this is these are the differences between RNA and DNA. So DNA has thymine base and with other bases which would be adenine, guanine, cytosine, and thymine. So it would have adenine, guanine, and cytosine, but in the place of thymine it would have uracil base. These are the two major differences between DNA and RNA. So now moving to the topic of DNA. DNA structure was so DNA structure was elucidated by Watson and Crick. These were the two scientists who predicted that, who found the structure of DNA. They found that the structure of DNA is that DNA is double helical structure. So this is the structure of DNA, and they have found the structure that DNA basically there are DNA is a double helical structure that is present in the cell and which actually contains contains the information for encoding the life. So it's a blueprint that encodes life. These are the four bases: adenine, thymine, uracil, guanine are the four bases which actually encode for the life. So Watson and Crick were the scientists who depicted the structure of DNA, and Erwin Chargaff was the scientist who established the idea of complementary pairing in DNA or RNA. Complementary base pairing in DNA and RNA. DNA is so here it established that adenine actually pairs with thymine. So adenine pair with two bonds pair with thymine. These are the hydrogen bonds. Okay. And guanine pairs with the cytosine. We remember that adenine is a purine base, thymine is a pyrimidine base. So here we know that guanine is actually a purine base, cytosine is actually a pyrimidine base. So guanine binds to three bonds with cytosine, adenine binds to two bonds with thymine. These are actually hydrogen bonds. In case of RNA, so adenine binds to uracil with two bonds, and while guanine binds at same cytosine. So there is no thymine in RNA. This is actually the structure. So this led to this prediction led to this discovery of DNA structure, which is double helical structure. DNA is a blueprint for the life, so it's a genetic material. So DNA has the capability to replicate itself. So this is all about the DNA. So we are going to discuss about the further about the DNA replication, how it does replicate in the next video tutorial, and we are going to discuss different types of RNA. Actually, there are three main types of RNA which are present in the cell. That is transfer RNA, messenger RNA, and the other is ribosomal RNA, which forms the actually part of ribosomes, and ribosomes help in the process of photosynthesis, protein synthesis. Protein synthesis in the process of translation. So we are going to discuss in detail about that. DNA structure was revealed by Watson and Crick. Erwin Chordoff predicted the complementary base pairing. Adenine binds to thymine with two hydrogen bonds. Guanine binds to cytosine with three hydrogen bonds. But this, in case of RNA, adenine binds to uracil, and it could be vice versa. Thymine might be present, it would bond to adenine, and cytosine might be present, which would bond to guanine. If you like the video, do comment, share, and like, and if do subscribe my channel for the further videos.